What if you could read the information inside our DNA? In 2003, the Human Genome Project was completed, taking more than a decade and relying on the efforts of scientists from different nations, resulting in the decryption of our species' genome sequence of more than 3 billion base pairs. Today, several other species have their genomes sequenced. This knowledge makes possible a series of advancements in science, medicine, and technology. The first DNA sequencing method, and the most used up to this day, was developed in the 70s by the biochemist Frederick Sanger and his collaborators. It is known as the Sanger sequencing method. Let's recall some basic principles of DNA. DNA is an organic molecule composed of basic units, called deoxyribonucleotides, that form chemical bonds with each other to build elongated chains. A deoxyribonucleotide is formed by a phosphate, a deoxyribose, and a nitrogenous base, which can be of 4 types: adenine, thymine, cytosine, and guanine. An elongated chain of deoxyribonucleotides linked together forms a single DNA strand, and 2 single strands join to form the DNA double helix molecule. To perform Sanger sequencing, we need a DNA sample, DNA polymerase enzyme, which will act in the production of a new strand of DNA, primers, which are simply a specific sequence of DNA that serve as a starting point for the DNA polymerase, deoxyribonucleotides, or DNTPs, and dideoxyribonucleotides, or DDNTPs. The principle of the technique lies in the structural difference between DNTP and DDNTP. While DNTP has a hydroxyl attached to the 3' carbon of the deoxyribose, this hydroxyl is absent in the DDNTP molecule. The presence of this hydroxyl is required for the chemical bonding between the units that make up the DNA chain. Because DDNTP does not have the hydroxyl in this position, it prevents a new DNTP molecule from binding, stopping the replication. At the beginning of the technique, a DNA sample is distributed evenly into 4 tubes, each containing the same amount of DNA polymerases, primers, DNTPs, and the same amount of a different DDNTP in each tube. These tubes will then be heated so that the double strands of DNA separate into single strands. The primer binds to a specific nucleotide sequence on the single strands, so that the DNA polymerase enzyme can start producing the new strand of DNA, inserting the nucleotides present in the solution. When, by chance, a DDNTP is inserted into the DNA strand by the enzyme, chain elongation stops. Thus, several DNA molecules of different sizes will be produced in each tube. Then, gel electrophoresis is performed, separating DNA molecules by their sizes. Because larger molecules are heavier, they run less on the gel, and the smaller molecules, being lighter, run more. Each column represents a DDNTP from a different tube, and each band shows the size of the DNA molecule with a specific DDNTP at the end of the chain. Thus, the result of electrophoresis informs us the sequence of the bases in the DNA strand of the sample, where the lowest band represents the shortest DDNTP interrupted strand, that is, the first base of the original DNA sequence, and so on, up to the uppermost band, representing the last base of the DNA sequence. With the help of this technique, we can study genes, look for DNA changes, diagnose disorders and anomalies in patients, unravel crimes, identify species, and better understand the history of evolution. By deciphering the letters of the genome, we perceive the differences and similarities between all living beings. We notice how each living being is unique, but at the same time possessing the same molecules shared by all organisms in our planet.